Ever stop and think about how a simple four-letter code in your DNA actually becomes, well, you? It's kind of mind-blowing, right? This is one of the most fundamental things in all of biology. It's like this tiny microscopic assembly line that's running non-stop in every single one of your cells. So let's dive in and check out this amazing molecular factory.
So yeah, that's the big question, isn't it? I mean, a blueprint is totally useless if there's nobody to read it and no factory to build the thing. So how in the world does a cell take this string of code, this molecule we call messenger RNA, or mRNA for short, and use it to build the actual proteins that make up, everything? Your muscles, your enzymes, all of it.
Well, the process is called translation. And honestly, the name is just perfect, because that is literally what's going on. The cell is translating from one chemical language, the language of nucleotides, which is what the mRNA blueprint is written in, into a totally different one, the language of amino acids. And those are the little building blocks that make up a protein.
Okay, so where this whole factory is set up, that actually depends on the cell. In complex cells, like ours, we call them eukaryotes, it's a super organized operation. Think of it like this. The DNA blueprint gets made in a protected main office, the nucleus. Then it's shipped out to the main factory floor, the cytoplasm, to get translated. But in simpler cells like bacteria, the prokaryotes, man, it's a whole different story. There are no walls. It's like a big open-plan workshop where the blueprint is being made and translated all at the same time in the same space. All right, so who are the key players on this factory floor? Before we see the assembly line in action, we've got to meet the molecular machinery that makes this whole thing possible. First up, the star of the show, the factory itself, the ribosome. This thing is an absolutely incredible molecular machine. It's where all the magic happens. It literally clamps down onto that mRNA blueprint and creates the perfect workspace for building the protein. And see those little slots? It has 3 super important docking bays, or sites, called the A, P, and E sites. And we're going to see what they do in just a minute. Next, you can't have a factory without delivery trucks, right? Well, meet the transfer RNA, or tRNA. You can think of each tRNA molecule as a specialized little delivery truck. Its one and only job is to go pick up a very specific building block, an amino acid, and haul it over to the ribosome factory at exactly the right moment.
So what are these building blocks they're delivering? They're amino acids. These are the raw materials for our product. And the whole point of translation, the entire goal, is to link these amino acids together in the exact order that the blueprint calls for. They get joined together by these really strong links called peptide bonds, forming this long chain, which then folds up into a working protein.
Okay, so we've got the factory, we've got the delivery trucks, and we've got the raw materials all lined up. But how does the factory know which delivery to ask for next? How does it know which amino acid to add? Well, it all comes down to decoding that genetic blueprint.
You see, the mRNA blueprint isn't read one letter at a time. It's read in little 3-letter words called codons. So a sequence of 3 nucleotides, like, say, AUG or GCC, that's a codon, and it calls for one specific amino acid. This chart here, it's basically the master dictionary. It's the Rosetta Stone that lets the cell translate from nucleotide speak to amino acid speak.
So how does the system make sure it gets the right amino acid every time? It's all about matching. Each of those tRNA delivery trucks has its own 3-letter sequence called an anticodon. Think of the codon on the blueprint as a lock, and the anticodon on the tRNA as the key. They have to match up perfectly. Only the tRNA with the right key can fit into the lock, and that's how you guarantee the right amino acid gets delivered. But here's a really cool thing. Nature is also super efficient. There's this idea called the wobble hypothesis, which was figured out by Francis Crick. It's basically a clever shortcut. It turns out that the match between the 3rd letter of the codon and the anticodon doesn't have to be quite so perfect. It can be a little, well, wobbly. And what this does is it lets a single tRNA delivery truck recognize a few different codons. This means the cell doesn't have to make as many different types of tRNAs. It's just a brilliant bit of biological engineering. All right, we've met the team, we understand the language of the blueprint, so now let's head down to the assembly line and watch a protein actually get built step by step. The whole assembly process really breaks down into 3 big stages. First, we have initiation, that's the setup. Then there's elongation, that's the actual building part. And finally, we've got termination, that's when the finished product gets released. So initiation is all about making sure we get started in exactly the right spot. the two halves of the ribosome factory, the small part and the big part. They come together and clamp onto the mRNA blueprint. Then, a special initiator tRNA, carrying the very first amino acid, locks onto the start codon. This is so important because it sets the reading frame for the entire rest of the message.
And here's a cool little detail for you. How does the ribosome in a bacterium know exactly where that start codon is? Well, just before the start codon, there's this special little sequence of letters called the Schindel-Garner sequence. It basically acts like a dock here sign, making sure the ribosome latches on in the perfect spot to get started.
All right, now for the main event, elongation. This is where the chain starts growing. With that first tRNA locked in, the next codon on the blueprint is now sitting in the ribosome's A site. So the right tRNA delivery truck zooms in, finds its match, and then, bam! The ribosome works its magic and forges a peptide bond, linking the new amino acid to the one before it.
And then the whole factory slides down the line. The ribosome lurches forward exactly one codon's worth down the mRNA. This awesome move is called translocation, and it shifts everything over one spot. The tRNA that's holding our growing protein chain is now in the P site. And, crucially, the A site is now wide open and ready for the next delivery. And to finish the cycle, the now empty tRNA, the one that just dropped off its cargo, gets kicked out through the E site. You can think of E for exit. And there we are, back at the start. The A site is free, ready for the next tRNA to come in. This whole cycle, a tRNA arrives, a bond forms, the ribosome moves, it just repeats over and over and over, building that protein chain at an incredible pace.
And this just keeps going, codon by codon, until a ribosome bumps into one of 3 special stop codons in the blueprint. Now, there are no tRNA trucks that match these codons. They're basically a big red sign that says the end. When the ribosome hits one of these, the whole assembly line grinds to a halt, and the final stage, termination, kicks in to release the brand new protein.
You know, when you take a step back and just think about this whole process, it's just breathtaking. This isn't just some random chemical reaction in a cell. This is quite literally the engine that drives all life as we know it.
So let's just do a quick recap of our whole molecular factory, okay? The blueprint, that's the mRNA. The factory itself, that's the ribosome. The delivery trucks are the tRNAs, and they're hauling the raw materials, the amino acids. And the finished product, a beautiful, perfectly built protein all ready to go out and do its job in the cell.
And don't think for a second that these factories are simple. I mean, just look at the scale of these things. The ribosome in a simple little E. coli bacterium is already this massive machine made of more than 50 different proteins. But the human version, it's even bigger and way more complex. We're talking 80 proteins, and it weighs almost double. These are seriously sophisticated pieces of natural machinery.
And you know what's really profound about that? The ribosome is what we call highly conserved. That's a fancy way of saying its basic design and how it works are almost the same across pretty much all life on Earth, from the simplest bacteria all the way up to us. This process is just so fundamental, so absolutely critical, that evolution has protected it for billions of years. It's a connection we share with almost every living thing.
So the next time you're just sitting there, think about this. Right now, inside of you, there are trillions, literally trillions, of these tiny molecular factories humming away. They're constantly reading your genetic code and building the very stuff that makes you you. It's happening right now. Which just leaves one amazing question. What incredible thing are they going to build next?